All right, folks, we're going to give something a try here as far as lab goes and see what happens. So, one of the things I wanted you guys to do, let me change colors here again. One of the things I wanted you guys to do in lab was to identify some bacteria um, based on a 16S R RNA gene sequence, right? And so, as you might can tell, the 16S rRNA gene sequence is a sequence in the genome of bacteria that codes for that 16S region in the ribosomal RNA. That region is pretty highly conserved among species because, again, remember, ribosomes make proteins. You don't want your ribosome you don't want the sequence in the DNA that makes the ribosomes to be altered very much because then you'll end up making faulty ribosomes that won't make proteins. That's not good, right? So that's a pretty highly conserved sequence. And so what we can do is um, if we have a stretch of DNA, I need to make sure my pad is straight here. Um, if we have a stretch of DNA, right? And in that stretch of DNA, a certain region of it is going to code for that 16S rRNA gene, right? And so because we know basically what that sequence is amongst different bacteria, we can create primers that'll sit down right, at that sequence, and they will amplify that whole region, right? And so we can run PCR on it, amplify it up, and then I can take that amplified product, right? Remember in those little bead tubes that we use to do PCR, I can take that amplified product and I can send it off and have it sequenced, and then I can get some information back. And then I can use that information to figure out what bacteria I have, right? So remember, I told you um, most of the time, so for us, what we did is in lab, we were growing bacteria up on plates, right? And so remember, I told you most of the time we cannot do that. Most bacteria cannot be grown that way. And so, in order to figure out what bacteria we have, one way is to do it this way, right? So, amplify a region of DNA, send that DNA off to be sequenced, and then we can compare that sequence with other known sequences and see what we have. And so, that's kind of what I want to show you today. I want to walk you through the process of how we were going to do that, right? And so, one of the things we were going to use is this company called Genewiz. They're the company that I would use to send samples off to actually have them sequenced. It can cost anywhere from $6 to $9, depending on what you want to have done. So for us, if we were going to do this in lab, it would probably cost about $9 a sample. Um, and so what I did early on is I sent a couple samples off just to give it a try and make sure this was going to work, right? And so what you can see is in Genewiz, and if you want to go to this website, you certainly can. Um, you're probably not going to be ordering anything, so you don't really need to know how to use this. But just as an example, um, what you can do at Genewiz is here I am looking at sequencing things. So we can sequence PCR product that is not purified, right? Which is what we would have ended up doing. Um, Now they actually offer the 16S rRNA gene sequencing themselves. So at GeneWiz, you can just send them a plate of bacteria, and they will pick colonies off and sequence them for you, right? And so we could have done that too. I didn't want to do that with you guys because that's just sending a plate off and getting some results back, and I wanted you guys to do some of the PCR yourselves. But here we are, right? Some standard RNA sequencing, some plasmid sequencing, right? GeneWiz does a lot of things, right? And so we would have just done this PCR um, purification process, and it'll let you walk through. I've been here before though, so let me skip all this, right? And so essentially, this is their order sheet, right? And so I think I have, let me see where my, up here, right? Oh, well, I can show you. Let me show you this first. So what you can do here is you can select what kind of DNA you want to send them, right? Are you sending them plasmids? Are you sending them purified PCR product? Are you sending them bacterial colonies? So we would have sent them unpurified PCR product, right? We could have told them the number of samples we're sending them, our order name, right? Tons of information we can give them here. Any information about the primers that we used, right? And so when we send that off, right, what we would have gotten back, um, let's see, I could show you, where are my, well, I'll just show you this. Right, so this is kind of what it would look like in the order form. Right, so unpurified PCR product, I gave it my initials as a name, bacteria 1, plus less than 500 base pairs. Ours probably would have been more than that. So on and so forth, right? And then you end up getting back a sequence, right? And that sequence will look something like this, right? And so it's just a string. Um, Now, there's a couple ways that you can get sequence data back. You can get it back as just raw data like this, or you can get them back in some different types of file extensions and things so that different programs can use them differently. But essentially, no matter how you get it back, the data itself is just a sequence of A's, C's, G's, and T's. Um, so remember, again, remember how we got here, right? So we started, right, with some bacteria. We didn't know what it was. We amplified a region of that 16S rRNA. We sent that amplified product off to Genewiz. They sequenced it, right? We still don't know what bacteria this is. So that's what we have to find out, right? So we'll take this sequence um, and we can put it in to some more software. And this is essentially what I'm going to want you to do for lab. So I've created 4 different sequences that I want you to work from, right? And so here's what you will do. Right? I want you to go to NCBI, right? So the National Center for Biotech Information, right? If you Google it like this, I'm going to want you to go to BLAST, but if you just click it, you'll also see BLAST over here at the side, right? So go to the NCBI website and go to BLAST, right? So BLAST stands for Basic Local Alignment Search Tool. And so the way to think about BLAST is you have a sequence, right? You have a DNA sequence, and you want to search amongst all of the known sequences that are out there to see how this lines up best. Sometimes your sequences will line up 100%. Sometimes they'll be 95%. Sometimes 75%, right? It just depends on how much work has been done with the bacteria that you are sequencing. Um, Maybe it doesn't line up with anything and you've got something brand new, right? So you just never know. And the way to think about BLAST is kind of like the Google for known sequences. And it doesn't have to be just bacteria, right? So when you're using BLAST, you could be working with humans, you could be working with any organism that has, well, any living organism, right? And so we're going to do nucleotide blasts, not protein blasts. So you'll click nucleotide blast, right? And then what I want you to do is this. I'm going to give you, right, several sequences, right? And all you have to do is this, right? You just copy this sequence. And to be truthful, you don't even have to copy the whole thing, right? So if I left off alpha C, that's fine, right? And so when I copy that sequence, what I want to do now is paste it into this box, right? And so there's all my A's, C's, T's, and G's, right, in the correct order, right? Um, and so here I'm doing a standard database search from the nucleotide collection, and I only want highly similar sequences, right? And so then I'll just click blast search. And it's going to take it a while, right? It might take it a minute or so. And so every time you see the screen flash, it's refreshing. And it'll tell you how often it's going to refresh, and it'll also tell you how long that it's been going, right? And so again, sometimes these can take uh, a bit of time to find, but you have to remember that sequence was probably 700 or more base pairs long, and you're trying to match it up base pair for base pair for any other sequence that's out there that comes close, right? And so that's why it takes a little bit of time. It's not quite as quick as a normal kind of Google search. But let's give it some time here and we'll see what's up. I've done this one before, so I know it'll come up. Sometimes it'll take 2 or 3 minutes. This one shouldn't take but maybe a minute, minute and a half. Or 2 minutes. It's amazing how long 27 seconds feels when you're waiting on it. And so here we go. Right? So the length, and this just gives you some basic information up here at the top. Like for instance, the length of our query was 799 base pairs. Um, but then, so here you can start to look down at some of these sequences and figure out what you've got. And so here it'll tell you kind of the query cover. And what I typically do is I look for the thing that repeats most, right? So if you notice up here at the top, we got this rhizobium, right? And that's listed as the top one. But then you see all these agrobacterium sequences, right? And you notice that all the agrobacteriums have 100% too. And so for me, I would say that the sequence that I gave you here was an Agrobacterium tumefaciens sequence because it just matches up with all of these different papers that have sequenced that particular um, part of the Agrobacterium DNA, right? And so what I want you to do is figure out which sequence that particular sequence matches up with, right? So which bacteria that particular sequence matches up with. And then I want you to look up that bacterium and see if you can find any kind of information about it. And just a quick little blurb, a few sentences about that bacterium is fine, right? Um, so not quite as robust as what I would want us to do actually in lab. This gets at some of the bioinformatics types of things that I think it's important to expose you to. Um, Something else you can do if you want to play around up here in these graphic summaries is, um, so let's say I only want to select that particular one, right? So I can select them all, right? Or just select the one. And then if I want to look at like alignments, so what alignments are is the top sequence is the query or the one that you're comparing it to, and the bottom sequence is the one that you uploaded, right? And so what you'll see is all of these match, right? So A to A, G to G. If there were no matches, right? And I think I can do this, right? So pairwise um, with dots for identities, right? So everywhere you see a dot, that's a match. If there was no match, then you would see um, a red letter, right? So for instance, if we got to right here, and if in the literature it says there should be a T there, but we had a G, then a G would show up down in this sequence and it would be red, right? And so that would be one difference that we might would have, right? Um, we can also do taxonomy information. So taxonomy is the study of naming things and the relationship that they have with things. Um, we're not so much worried about that right now, though. That would be more of a maybe a Bio 112 type of thing, right? But that's what I want you to do. I'm going to give you a sequence, right? I want you to blast that sequence, and then tell me what species you come up with, and then give me some information about that species. Again, to get to blast, you go to NCBI, and then you'll click on the blast over on the right-hand side. All right, so if you have any questions about this, shoot me an email and we can discuss it. Or however you want to get a hold of me, I don't care at this point. If you want to message me through I mean, I have students send me messages through Facebook Messenger all the time. I'm fine with that, especially now. However you want to get a hold of me, get a hold of me. Um, it doesn't matter to me whatsoever.